Hi, hello everyone. I'm Sayali, and I'm a PhD student at the University of Wisconsin-Madison. I work with Professor Daifeng Wang at the Weizmann Center, and today I will be talking about our recently published paper called CMOT, short for Cross-Modality Optimal Transport for Multimodal Inference. In today's talk, I will go over some background information, literature review, then I'll discuss our method, and followed by some discussion and future work for this method. Oh. So with next generation sequencing technologies, we are able to capture molecular activities inside a single cell, which means we have multiomics data for each cell characterizing their molecular activities. For example, at the genome level, we have DNA data. At the epigenome, we have chromatin accessibility and so on. Um, and amongst these, we focus on epigenome and transcriptome mostly because not only they significantly affect the cellular functions in our body, but epigenomes are known to influence transcriptomes through gene regulation. And therefore, we focus more on these two activities and more, especially on the joint analysis of these two. As an example, why joint analysis is important, a recent study was able to annotate cell type clusters in chromatin accessibility by doing a joint analysis of both gene expression and chromatin accessibility. Also, new sequencing technologies have been able to jointly profile multiple modalities from the same cells. Such profiling experiments have made it possible to simultaneously study different modalities from the same cells. But these experiments are not easy, especially because they're expensive. They have low recovery of cells, and the data collected is sparse and noisy. And so, therefore, um, oh, sorry. And so joint profiling is not done as commonly as opposed to existing single profiling methods like single cell attack-seq or single cell RNA-seq, which means we have more single modality data compared to joint modalities. So this creates a space for methods that can utilize these single modalities and impute the missing ones. And so several methods have been developed in the recent years to infer missing modalities so we can do the joint analysis of the cells. I will briefly review some existing methods and their limitations that we are trying to address in our method. Here I look at 4 state-of-art methods, Surat, MUFA Plus, GLUE, and Total VI that have been developed for integration and modality imputation in single cells. And the limitations that these methods pose is that most of them need jointly profiled data for training. They have certain assumptions about underlying distributions of modalities, or they're designed for specific modalities, or they need some kind of prior biological knowledge between genes and peaks, for example. So, to address these limitations, we have developed an approach that uses optimal transport and manifold alignment. So, CMOT infers missing modalities by combining manifold alignment and optimal transport. And it takes two training modalities X and Y and a test modality Y hat as inputs, and infers the missing modality X hat for the cells in Y hat with the assumption that Y and Y hat share the same features and X and X hat share the same features. And also, if available, it accepts an optional input about prior knowledge about cells in Y, which could be like cell type labels, for example. Here is a detailed flowchart of the method divided into 3 steps, where step A is manifold alignment of cells in X and Y if they're not jointly profiled. So this step is optional if you have a joint jointly profiled training data set. Step B is optimal transport, where we find an efficient transport plan between cells of Y and Y hat. And in step C, uh, we use the k nearest neighbors to infer X hat profile for cells in Y hat. Going into step A, where if the cells in X and Y are not aligned, we first align them using manifold alignment so that they're brought to the same space. And after this, the cells in both modalities can be matched using Euclidean distance, for example. In particular, we optimize this following equation that tries to learn an embedding or a mapping function phi x star and phi y star at the same time. So this optimization tries to preserve the local geometry of each modality, and at the same time, it matches any partial cell-to-cell correspondence between X and Y. In step B, we apply optimal transport to modalities Y and Y hat that have similar features, and we bring them to the same space. So here we optimize this equation to find the most efficient transport plan called pi star that helps move the cells from Y space to the cells in Y hat space. So this optimization problem tries to minimize the cost of transportation between the cells of Y and Y hat, further regularized by an entropy for a diffuse transportation plan and and some prior knowledge if we have about the cells in Y hat. So on solving this optimization problem, we get a probabilistic mapping or a coupling plan that tells us how much of a cell from Y must map to a cell in Y hat space. So this, which can be used to transport the cells from Y to Y hat. And finally, with the help of, in step C, with the help of aligned cells from step A and the transported cells from step B, we can infer the missing modality X hat using a weighted k-nearest neighbor. And here for every cell in Y hat, we look for k-nearest neighbors of this cell amongst the transported cells from Y. Then we look at the gene expressions of these transported cells from step A and perform a weighted average of these cells. Okay, next I'll talk about the results for missing modality inference in one of the applications. We applied the method to 4 data sets, but today I'll talk about the first one, which was an application to a developing human brain, which had approximately 8,000 cells, where X modality was gene expression and Y was chromatin accessibility. So in this application, our aim was to predict gene expression for cells which only have chromatin accessibility information. So we divided the data set of 8,000 cells into 80% training set and held out 20% for the testing. We normalized these data sets and chose 1,000 most variable features from each. So we did all the training and cross-validation on the 80% set and evaluated on the 20% held out cells. As part of evaluations, we did cell-wise and gene-wise Pearson correlation. And we also calculated an AUPRC score for the known cell type marker genes. Okay, coming to training and evaluation, this method can be tuned for 5 parameters. The first is the choice of the latent dimension that we want to use for step A, which is manifold alignment. So the first graph shows the evaluation of how we choose D for the alignment, which is evaluated using the score called fraction of samples closer than true match. It measures how close a cell from one data set is matched to its corresponding cell in another data set. So the lower the score, the better is the alignment performance. Here we chose 20 because after 20, we don't see much difference in the alignment performance. Um, for step B, which is the optimal transport step, we had two regularization coefficients, lambda and eta, which were again chosen based on cross-validation inference. Sorry. Um, and for step C, where we had to choose k nearest neighbors and how many features we wanted to use, again, we tested this for multiple number of neighbors and multiple number of highly variable features from the data set. And if you can see the graph on the right, that is the one that shows that at k equal to 600 and for 1000 most variable features, the performance almost plateaus and gives the highest correlation value. So we chose that. Here are the results of imputation. The first four scatter plots show the performance of imputation through regression plots. Each plot represents one cell from a different cell type, where y-axis is the inferred expression by the method, and x-axis is the actual measured gene expression value. And we see that each of the cells shows a high correlation value. And on the right side, we show the performance. The right plot shows the AUPRC of the marker genes from each cell type based on the inferred expression. So marker genes are usually highly expressed in their respective cell types. Therefore, their gene expression values are relatively higher than the rest of the genes. So a higher AUPRC shows that these genes were highly expressed in these cell types based on the inferred expression. Next, we want to show the comparison with other methods. So the first two scatter plots show the gene-wise Pearson correlation values for different genes in the data set. The numbers inside these plots show how many genes had a higher correlation value with respect to that method. So we showed this for two popular methods, SURAT and MOFA+, and CMOT was able to infer more genes with a better correlation than the other two. The Weiland plot on the right shows the cell-wise Pearson correlation values between the inferred and the measured expression. So we tested CMOT for different levels of correlation between the training modalities X and Y, ranging from 25% to 100% correspondence between the X and Y cells. And overall, we see that the median correlation of CMOT is higher than most methods, as well as the computation time is significantly lower. Next, I'll move on to the discussion in future work for this project. To summarize, CMOT is a computational framework that can infer missing modalities for cells that only have one modality. And this is particularly important if you wanted to do an integrative analysis. Some limitations addressed by CMOT, by limitations of existing methods, are that it can be extended to different modalities like protein expressions. It does not need to be trained on 100% jointly profiled cells. It is computationally efficient and faster. Also, flexible framework where step A, which is the alignment, can easily be replaced with other methods like CCA. However, there are a couple of limitations. Some of them are that the performance scales quadratically with the data sets. This is because manifold alignment performance scales quadratically. So if we wanted to uh, uh, go work around with that, we could use other alignment methods. Another limitation is the conservative mass balancing property of optimal transport, which means that each cell in the source must be matched to the target, which is not always true because real world data sets are generated differently. So we wanted to handle that by using emerging approaches like unbalanced optimal transport. And moving on to some future work for this project, we want to expand this to different OT variants like Romo Wasserstein distance, so that we can directly apply this method to two different modalities. or also include more modalities like cell morphologies or electrophysiological data, and also extend this to an additional functionality where we can predict labels for the cells in Y hat. We are also working on extending CMOT for spatial imputation by incorporating autoencoders and optimal transport. Um, our lab at UW Madison recently built a flow for single cell multimodality modeling, from building a multi-view framework extending to methods for multimodal alignment, cross-modality imputation, and translation. Yes. And I want to thank all my amazing lab members and Professor Wang, and we can move on to questions.
Thanks very much. Time for one question, one quick question. Any questions? Okay, I have a question about more about future directions. So the alignment step.
Yes.
Is there a way to capture uncertainty in that step and use it in the next step? Meaning that, you know, if there are parts of the cells that are unlikely to be well aligned, could you deal with them differently in the OT step?
Yes. Actually, I did not put it in the slides, but we have a check in the method that filters out cells that are not properly aligned. So we, instead of imputing wrong expression, we simply remove them. So that's a better way to handle it.
All right. Thank you.